Last class we talked about carbohydrates, right? Are there any questions on carbohydrates, glycosidic linkages, what they are? That video is also up on Moodle, so you feel free to go through that. And if you have questions, come see me. All right, I know a couple of students from other classes came to see me about the other sections, came to see me about glycosidic linkages, and there was some confusion there, so I wanted to make sure that everyone here, good. Difference between starch, cellulose, glycogen, and their linkages and what they mean. All right? Any other questions about carbohydrates in general? All right. Today we're going to go through, hopefully get through lipids and proteins before we take the quiz. All right? That means we're going to go pretty fast. Oh, I know. But it's really not that bad. Trust me. Okay? We're going to go fast through slides. Just listen, and you can go back with the lecture as it's being videotaped. All right? First off, lipids are a macromolecule. Okay? It's diverse in terms of their structures. All right? In this case, we've been talking about macromolecules and they form polymers. Lipids really don't form true polymers. They are formed from subunits, but not like in carbohydrates where you link one polymer of glucose or one monomer of glucose to another, to a third, to a fourth, and so on. In this case, it's sort of one subunit A plus subunit B that are different are put together. All right, and then you don't link any more subunits on top of that. All right? But there are things that lipids are composed of. All right? They have little or no affinity for water. They are hydrophobic. Okay? They are almost consistently made up of hydrocarbons, carbons bound to a hydrogen, and for the most part, you see completely non-polar covalent bonds. That is the property of the molecule that gives it its hydrophobicity. Okay? Because we have a non-polar covalent bond, which means what? There's no charge. Non-polar means how are the electrons shared? Equally. Right? So then there's no charge. All right? And then that is directly related to its fact that it's non-polar covalent bonds. All right? And we're going to talk about fats, phospholipids, and steroids. Okay? First off, fats. Two types of fats, or there are two types of fats. We'll get to that in a second. But fats are composed from two different subunits. They are composed from a glycerol molecule, which is a 3-carbon sugar, okay, a carbohydrate, and fatty acids. All right? So a fatty acid is a long chain of nonpolar carbons bound together, and then you end up at the end of that, you have a carboxylic acid group, which you've seen in your chart of your functional groups. It's C, double bond O, and then OH off the other one. So it looks like this. So you have carbon, 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 and then this carbon has a double bond O, OH. Right? This is the carboxyl group attached to your long carbon skeleton. Right? That's what a fatty acid looks like. Right? Glycerol is a 3-carbon molecule, right? And then it has oxygens in between that and where the fatty acids are going to link to. All right? And there's a better picture on the next bit. Okay? This is glycerol and your fatty acids. All right? How are we going to build this fat? This fat we're going to call the triglyceride. Why a triglyceride? Tri refers to the number 3. Glycer refers to what? Glyceride refers to glycerol. 3 fatty acids on glycerol. That's all triglyceride means. All right? So we're going to form this triacylglycerol. How are we going to form it? How are we going to synthesize this molecule? Dehydration synthesis. So let's look at this. Where are the components of dehydration synthesis going to occur? We have to take out water. So where are we going to get water from? The OH here and the hydrogen there. Okay. Either way, if you do it one way or the other, take a hydrogen off the other, in whatever way it's going to happen, there's going to be an oxygen left over, right? So water's going to come out, and this is going to happen on all 3 of these. H2O is going to come out, and you're going to end up with this. Say this comes off, this carbon's going to link to this oxygen, right? That carbon's going to link to that oxygen, and so forth. Now that fatty acid chain is going to be linked to the glycerol through a D. This is what it looks like. In this case, it's palmitic acid that you're looking at. All right, the C double bond OH. OH comes off. You end up with the CO. Right, that's what's called that bond between the glycerol and the fatty acid is called an ester linkage. It's called an ester linkage because of the COC, which is bound to. That second carbon is bonded to a double bond oxygen. Right, that's an ester linkage. You end up with your fat molecule, or triacylglycerol, triglyceride, same thing. Right? Questions on that? That is one type of, that is fats. That's how fats are, triglycerides are made. One type of lipid. Right? So how do we get different types of fats? How do we get different types of triacylglycerol? Like you have palmitic acid up there. What's the difference? What can I change that's going to change this fat? You can, depending on which type of fatty acid subunit you use, that's going to change the type of fat that it is. Okay? So your fatty acids then, they vary in length. Okay? They vary in the number and locations of double bonds. Some of them have bonds, some double bonds, some of them do not. If they have double bonds, right, they are called unsaturated fats. If they have no double bonds, they are saturated. What are the carbons saturated with if there are no double bonds? So here's a carbon chain. Right? Each of these carbons has 4 possible groups. What are they saturated with? These lines are actually going to bond to what? They symbolize a bond to what? Hydrogen. They're just not written there. So these carbons are saturated with hydrogen. Now, if I make a double bond between these 2 carbons, right? If you count the number of bonds each of those are making, it's 5. So we have to remove one of them. Now, are all those carbons saturated with hydrogen? No. Right? You could put more hydrogens in there if you broke that double bond. That's the distinction between a saturated fat and an unsaturated fatty acid. All right? So a saturated fatty acid, the fatty acid bonds are all single bonds. Unsaturated fatty acid, at least one or more double bonds are present. A saturated fat has a saturated fatty acid, and an unsaturated fat has at least one of those triglycerides, or those fatty acids in there, is an unsaturated fatty acid. Okay. Questions on saturated versus unsaturated fatty acids and fats? All right. Saturated fats are mainly animal-derived. Okay. There's your structure of fatty acid molecule, you see here. All right. And your space-filling molecule. The other thing about double bonds is that the presence of double bonds bends the shape of the fatty acid. Because it bends the shape, it sort of kinks the molecule. It prevents the lipids from packing in too closely. Okay? Because they don't pack in so closely, you end up with oils, which are unsaturated fats that are liquid at room temperature. Right? And that's directly related to the packing in of the lipids. All right? So a saturated fat, solid at room temperature, no double bonds. An unsaturated fat, liquid at room temperature, mainly plant-derived, and they are the presence of double bonds. Questions? Straightforward, right? Other type, second type of lipid, phospholipids. All right? Little different, still the same subunits. Glycerol, fatty acids, but instead of 3 fatty acids within a triglyceride, we only have 2. So we switch out one of the fatty acid chains for something else. What is that something else? A phosphate group. So we're still going to have our glycerol molecule, 1, 2, 3, right? O, right? And then your fatty acid side chain, but instead we're going to add this to be a PO4. Okay? That's the basic structure of a phospholipid. You'll see a better picture of it in a second. Okay? Fatty acids are nonpolar, correct? They're hydrophobic, right? What about the phosphate? It's hydrophilic. Why is it hydrophilic? Because it has a charge, right? It has a charge. It has a slight positive charge. All right? Because it has the charge, it tends to like water. Right? No charge, doesn't like water, so it separates between the two. All right? So you have what we call the hydrophilic head and the hydrophobic tails. Lipid, right, which they show here. Okay? So you have a choline. This is phospholipid is phosphatidylcholine. Right? Your structural formula giving the presence of a double bond in the fatty acid. Right? And there are different types of phospholipids. Some have double bonds, some do not have double bonds. All right? In this case, this one does. You have instead of having the glycerol or the phosphate attached to the middle carbon of the glycerol, it's on one end. Doesn't matter, right? But these are all different ways of representing phospholipids. Okay? Basically hydrophilic head, hydrophobic tails. And when placed in water, spontaneously phospholipids form a bilayer. Why?
Because the hydrophobic tails don't really be near the water, so they fall like away from it.
So the tails arrange themselves. So if this is water, right? Water everywhere. The nonpolar regions, the hydrophobic regions, will arrange themselves like that. And so you have your phospholipid bilayer. That spontaneously occurs when phospholipids are put in water and is thought to be the evolutionary step which occurred for the formation of primitive membranes. So that this is basically saying the same thing. This feature of phospholipids is a feature of cell arrangement found in cell membranes. Right? And depending upon the arrangement of phospholipids, what fatty acids are there, changes properties of a cell membrane. So you get a difference in terms of cell membrane of a paramecium or an archaea prokaryote versus human cells. Right? That has to do with the constituents of the phospholipid bilayer. Right? Our third type of fat are fats, steroids, or third type of lipids, sorry. Steroids. Steroids are lipids characterized by a carbon skeleton that it has a distinct structure. They're consisted of 4 fused rings. All right, 3 6-member rings, and then 1 5-member ring. All right, so 1, 2, 3, and this is your 5. Okay, that's the basic structure of a steroid. All right, now a difference between, and this is also the basic structure of what steroids use, or the starting material that's used to produce steroids, cholesterol. Okay, now give me examples of steroids. Testosterone. What else? Estrogen. Anything else? Pregnenolone, progesterone. There's a whole bunch of synthetic ones that you can go. DHEA. What? Prednisone is also a steroid. It's not a derivative of this. It's a synthetic one. Okay. Glucocorticoids such as cortisol. You have thyroid hormone, are all based upon this structure. Okay. And in the formation of these steroids and how they're produced, what are you going to do what would you do if you were going to ask as a organic chemist to produce a synthetic form of this steroid? You're going to start with cholesterol, and then what? How does the body make testosterone from a cholesterol background? What differentiates testosterone from estrogen if they have the same background? They have the same basic structure. Somewhat, the arrangement of double bonds, but this structure is going to be the same. What the side groups off of this, the functional groups that come off of this, will change. All right, so if I added double bond O here, or a chain of carbons up there, add a carbon here, or change the constituency of that ring. When I change the side groups, or what we call the functional groups, that changes the interaction that they have. All right, so if that's a double bond O, or if it's a OH, that's one of the big differences between estrogen and testosterone. All right, then this also makes this a benzene ring instead of a presence of double bonds in the ring structure. That changes its function. Okay, so here's cholesterol. Okay, here's one of the functional groups. Here's another big functional group, and these will also change. All right, you will also get arrangements of double bonds in here. All right, but the big thing in terms of the function, you still have your basic structure, your 3 6-member rings and a 5-member ring, but arrangements of the functional groups on the side are. Questions on steroids? Okay. Questions on lipids in general? All right. The next macromolecule, and the most diverse in terms of function and most important in terms of structure, are proteins. Okay? Proteins are composed of long chains of amino acids. So what is the subunit of carbohydrates? Monosaccharides. What's the subunits of lipids? Glycerol and fatty acids for lipids, or for fats, and phospholipids. What's the subunit or the starting material for steroids? Cholesterol. Okay? And what's the starting material, the subunits for proteins? Amino acids. Okay? Proteins have diverse functions. They include defense, storage, transport, cellular communication, movement, and structural support. All right, so they are by far the most diverse in terms of functionality, all right, of the macromolecules. And the next 2 slides shows of their diversity. So you have enzymatic proteins, enzymes, defensive proteins such as antibodies. You have storage proteins such as casein, the protein of milk, albumin. You have transport proteins. You have integral membrane proteins allow transport between one side and the other. An ATPase pump is one of them, sodium potassium pump for neurons and muscle. You have hormonal proteins such as insulin. It's a protein that's produced in the pancreas, transports throughout the body, right, and elicits some type of response in other cells. With that, there's a receptor. Proteins are receptors as well, so they will recognize and bind to hormones or other proteins and elicit some type of response within the cell. Contractile motor proteins, muscle contraction is directed based upon actin-myosin binding and contraction. And you have structural proteins such as collagen and elastin, which help for support for the body. So, complete diversity, right? And that's not to say that collagen or actin and myosin they only do their one function, because they actually can function in multiple ways. Okay? Questions on functions of proteins. So, enzymes, which are important. Life would not be possible without enzymes, because all the reactions that occur in our body do not necessarily occur spontaneously, right? At least not in ambient temperatures. So we need enzymes to allow those reactions to occur, all right? So they act as catalysts by lowering activation energy, and that's a chemistry class, not a biology class. Know that they are biological catalysts, and they allow chemical reactions to occur within the body. All right? So, amino acids. Okay? We're going to get to what an amino acid is in a second and what they are. But proteins are different. This is a little bit of a language difference. Okay? So, a protein that we talk about, that I will use, will say protein distinguishes functionality. Okay? If I say protein, that means a functional unit that is different from a peptide or a polypeptide. All right? A peptide, what is a peptide? What are peptides? Bonds. Okay? And so a peptide is a bond between two amino acids to form a polymer, which will end up forming a functional protein at some point. Okay? But, so a polypeptide is really a long chain of amino acids put together. The difference between a polypeptide and a protein is functionality. Okay? Protein denotes complete function. A polypeptide may or may not be a completely functional protein. Okay? So, polypeptide, all proteins are polypeptides, but not all polypeptides are proteins. Understand the distinction? All right? It's just a language thing. For the most part, I will use the term protein as understand that means a functional unit. All right? So, amino acids, which are the subunits of proteins. They have a distinct structure. Okay? They differ. They are all the same. Every amino acid, all 20 of them, have something in common. Okay? They are built around a central carbon. And off that carbon come certain groups. You have an amine group, which is NH2. Okay? And on the other end, you have a carboxylic acid group. So you have a carboxyl group, a C, double bond O, OH. All right? So this is your amine group. Here's your carboxylic acid group. And then a hydrogen. All right? And then you get the difference. This is the group that is important, the R group. Okay? That is the difference between tryptophan, and glycine. And glycine has a different R group than alanine. And alanine has a different R group than isoleucine. Okay? It can be as simple as a hydrogen, as complex as a ring structure. Okay? Doesn't matter. All you need to know is that the R group distinguishes one amino acid from another. No, you do not need to know what those R groups are. Okay? You do not need to know the structure of every amino acid. All right? Know that the difference is in the R group. Okay? So here are your 20 amino acids. All right? Look at the difference as complex or as simple as a hydrogen to as complex as a fused ring structure, another ring structure. All right? So you have tryptophan versus tyrosine. Okay? The R groups also give that amino acid its functionality. Some of them are nonpolar. Some of them are polar. Some of them are acidic. Some of them are basic. Why do you think that's important? Why are the R groups important and what their properties are important? Sabrina?
Proteins are, and their function
All right, so,
it has to, it's going to be more acidic in certain parts of the body.
okay.
That's basic.
All right, so I think what you're trying to say is that the protein is going, the property of the protein is going to depend on the sequence of amino acids and the sequence of the properties of those amino acids. The properties of those amino acids depend upon this, upon these structures. Okay? And the integration, what R groups are where, will also help to determine protein function and structure. All right? And we'll see that on Monday. Okay? Oh, Wednesday. Sorry, Wednesday. All right? Questions on protein, on amino acids and proteins up to there? No? Yes? All right.